In the last video, we talked about the electrical impulse. So that is polarization, depolarization, and repolarization, and what's happening to the ions as they move across the membrane of a neuron. Now, this is happening similar to a domino effect, where we're only looking at one area at a time, sort of in that example with those segments, but it's really a process that occurs along the entire membrane of a neuron. There is a very short period of time after repolarization occurs that a refractory period occurs. And that refractory period is a moment where that neuron is trying to make sure that the membrane is at a polarized state again so that another action potential can come through if that neuron gets stimulated again. So we sometimes represent this in a graph. And so you can see there's two graphs here, a very simple one on the left and a little bit more annotated one on the right. On the one on the left, we can look at each little segment of that graph. So if we were to imagine a giant squid axon, and we've attached a computer or voltmeter to that giant squid axon so that we can measure what's happening to the nerve membrane as it gets stimulated. So before that neuron gets stimulated, it's polarized. So that's this section here that I'm going to highlight in yellow. So that's the polarized area. That's when that neuron is at rest. Nothing is bothering it. And that's sort of that potential energy that it has to be able to act if a stimulus hits it. So the stimulus in this case would be acting right there, right where you can see that graph start to increase. The area of increase, that's going to be the area where depolarization is occurring. So depolarization, remember from the previous page, we have sodium rushing into the neuron, and when that positive charge rushes into the neuron by diffusion, it's going to increase the charge in the neuron, making it more positive. And you can see by this graph, it's showing that it's going up to about 30. So it's 30 or 40 typically is what we say. Then once all the sodium has come in and your sodium channels have completely closed, then repolarization needs to take place. So I'll sort of give that a little bit of a highlight in blue. Repolarization is where the potassium gates open and potassium starts to diffuse out of the neuron to try to make that axon more negative at that point again, right? We want to make it polarized. We're repolarizing it. Now that's only going to go so far, as we saw on the previous page, because you don't have really enough enough potassium to change the potential back to -70. It only goes to about -50, -55. And you can see even in this example here where I've highlighted in blue, I didn't actually take it all the way down. Because what's going to happen there is in the bottom part of this, no more potassium can move. So this is where the ATP pump really has to kick in to gear to actually use energy or active transport and ATP to pull that sodium out of the neuron and put that potassium back into the neuron. And so really your refractory and your repolarization are kind of one and the same. We're trying to repolarize the cell. The repolarization is occurring when the potassium ions are trying to repolarize. And then when the cell realizes, you know what, I just got to use energy to make this happen, and the ATP pump really starts to kick in, that's that area that's highlighted in sort of that pinkish red color. The other thing you're going to notice too is you can see that at this little point between here and here, that dips down below the -70 value. That is an area of hyperpolarization. And when it's hyperpolarized at that point, it's around -90 millivolts. And the reason that happens is if we think about what's happening with the sodium and the potassium, we're at the point where we've got some sodium on the inside, and of course we've got some potassium, and some potassium on the outside, and then some sodium on the outside. When the ATP pump comes in and says, okay, I'm going to pull the sodium out, those sodium gates that the sodium had gone through to go in, they were shut. They were shut a long time ago at the end of depolarization. But the potassium gates, remember, they actually stay open and they close very, very slowly. And so when the ATP pump takes this sodium out, that's fine. The sodium can't diffuse back in. But when it kicks or pulls the potassium in from the outside, so it's going to grab this potassium here and pull it in, this door is still closing. And with this door still closing, this potassium then can sneak back out the door. And so there's this moment where you're actually losing a lot of positive charge from the inside. Not only are the sodiums going out, and the potassiums are trying to go in, but they keep sneaking out because this door closes very, very slowly. This channel, that potassium gate, closes much slower. It finally closes around, somewhere around here, or on this graph, it would be, it's a little bit different on this graph, but somewhere where you can actually start to see some progress of that cell becoming around -70 for polarized. So they do close eventually, but because they take longer, we get this period of hyperpolarization that exists. The next graph on here shows you when the sodium channels are opening and when they're closing, and when the potassium channels are opening or closing, and that's a good diagram to show you. So we've got a point where the stimulus occurs right here. We've got some sodium channels opening in this yellow section, and then you've got more sodium channels opening. There's actually a little point in here where, and I'll try to draw it straight, where you can see that the slope of this line changes between the yellow section and the green section. What happens initially when something is a stimulus onto a nerve, it only just lets a few sodium in. And there has to be enough sodium that goes in that's actually going to cause a significant change in the membrane potential. And that line there, and you can see it's labeled over here, that's called your threshold level. And the threshold level usually sits around -55 for our typical type of neuron that we talk about. So when we're looking at a particular graph, you'll always see the line going up just with a smaller slope, like in the yellow section here, and then a greater slope if it's going to have a full action potential. And I'll talk about that as we get into the last little section of this page. So the different speed of nerve impulses can be based on where they are in their body, the axon diameter, but what we really focus on in Bio30 is whether or not they're myelinated or unmyelinated. Myelinated neurons have a faster transmission speed than unmyelinated neurons. So if you think about from the same sort of length of a neuron, and I've got this polarized area inside, you've got all this membrane where you're going to have this, sodium's going in, potassium's going out, and so on. But if you have a myelinated neuron, everywhere the myelin is covering, you're not having any action potential happen at that point, because these ions are soluble and the Schwann cells are very lipid rich. So the only places that the impulse can go in are at these sections. So there's a lot less sort of sodium movement along the surface area of the membrane in this example over here, and that makes this happen a lot faster than this one. The all or none response is running back to that idea of this threshold. They're kind of interchangeable, they overlap a little bit in terms of their definition. But a stimulation or a stimulus must be large enough to start an impulse. So it has to reach a threshold level. And if it doesn't, then there would be no response. So if we were to, in very simple terms, just sketch one of these little action potential graphs, if you have an all response, and I'll draw a little threshold line in here. If you have an all response, so you got a polarization, it goes up, it reaches threshold, and then we get this action potential that happens. This would be my all. None would be, there just isn't quite enough stimulation on that nerve to actually reach that threshold, and so it just drops, and that nerve or that segment of the nerve just stays at a polarized state. This is the case where it's like the mosquito lands on you and it bites you and you feel it. You get it fully to its potential. And the other example over here is where the mosquito just doesn't hit the nerves enough, right? It's walking really lightly on the skin and you don't feel it, and it didn't send an action potential to tell you that something was on you. So that's this idea of the all or none response. So the all or none is that the nerve is either going to fire in the case of example 1, or it's not going to fire in case of example 2, and that's all based on what the threshold level of that neuron is. The idea of summation is that sometimes more than one nerve, or neuron we could say, because that's how we're sort of looking at them, is required to stimulate another neuron. And we're going to get into how these things work, how these neurons work along a pathway. But if I had, two neurons here, there's their axons and their axon terminals. And those axon terminals are in very close contact with a third. So basically what this is saying here is that I might need stimulation. These are chemical transmitters. I might need stimulation from A and B in order for C to meet its threshold so that it can actually send a message. If I only had neuron B stimulating, and let's say A wasn't doing anything, just those 4 chemical transmitters might not be enough for C to have enough to reach its threshold to actually send a nerve impulse down it. To kind of put some of those things together before we finish off the electrical impulse is thinking of what happens when the intensity of your nerves, or whatever the feeling you get from your nerve being stimulated, changes. So we're going to look at something like, if somebody taps your shoulder, a touch, versus somebody punches your shoulder, right? So if somebody touches your shoulder, we're going to draw these graphs. So this is my membrane potential in millivolts. Same over here. And it's going to be over time as our x-axis in milliseconds. And I'll draw a threshold in here, and I'm going to try to make these comparable by putting at the same point. So if this is my touch graph, well, I'm going to have a polarized state, and then it's going to go up a little bit, and then it's going to go up very significantly, and that was not very straight. A little harder to do on the iPad than it would be on paper. And then back down, a little bit dipping, and then so on. So this would be at a level of -70. This would be, let's say, +40 millivolts. That would be a touch. What a punch is, and some people would think, oh, well, you're going to go way above 40. That's not what it is. The intensity of the stimulus is determined or being communicated by the neuron by increased frequency. So how that's going to look, and this is a little harder to draw, is going to go up and then up and down. And you're going to get more waves in a particular period of time. So this is increased frequency. So in the same time it took to have one impulse in touch, I was fitting about 3 in there. It doesn't go any higher. It still only goes to +40. It's all or none. It's just going to increase the frequency by which it happens. So it's not like if I drew a 4th graph on here and I was to draw it, it's not like this thing goes way higher than 40, right? Where 40 is here, and then it goes higher than that. No, that's not what happens. Okay, it actually just increases frequency. You're going to get more cycles per second, is essentially what that is.